Okay, so in chapter 13, we're going to be talking about characterizing and classifying viruses, viroids, and prions. And this is probably one of my favorite microbiology topics and a major concentration of what I studied in graduate school for my master's. Okay, so viruses are really different from the prokaryotic and eukaryotic cells that we talked about previously in this course because they are acellular. So that means that they don't have a cytoplasmic membrane, they don't have cytosol or ribosomes or other organelles. Instead, all they really are just nucleic acid. So that nucleic acid can be either DNA or RNA, but never both. And then that nucleic acid is just going to be surrounded by a protein coat. And we call that protein coat a capsid. So some viruses do have an additional layer on the outside, so they could potentially have an outer phospholipid envelope. And if they have one, they really just got it from the host cell membrane when they were lysing the host itself. Downside is that viruses that do have this outer phospholipid envelope will help them evade host immune responses, and it can also help them attach to new host cells. Again, if you remember, viruses, they lack the machinery that's needed for a lot of processes. So like things like energy production, metabolism, or protein synthesis, they don't have those organelles and can't undergo those independently or by themselves, including replication. So in order to replicate, they have to invade a host cell, hijack its host cell machinery. So things involved in like metabolic pathways or those involved in making other viral components. And this is what really makes them different from all the other microorganisms that we've talked about. So viruses exhibit two different states in their life cycle. So they either have an extracellular state or an intracellular state. And in the extracellular state, the virus is considered to be a virion. And a virion is just a complete infectious particle containing its genome, so that DNA or the RNA, surrounded by that protein capsid, and then if it has an envelope as well. When it's in this extracellular state, the virus is considered to be inert, so it's not reactive. It can't move, it can't replicate, and it can't respond to its environment. However, once it enters a host cell, the virus can kind of shed its capsid or its envelope and release its genetic material into the host cell. Once it's inside, it's considered to be in the intracellular state. And the virus is no longer a complete virion, but just nucleic acid. And then it's going to use the host enzymes and ribosomes and energy to help replicate and assemble new virions. So it kind of has this dual nature, one where it's completely inactive outside of the cell, and then one where it's inside of the cell and able to like use the host cell machinery to get things done. This is just showing you kind of an image of what a typical virus looks like. So this tan stuff right here is this whole thing is just representing the capsid that's made up of just proteins. And then inside, in this case, this is pretty much just the simplest type of virus we could have. It's just nucleic acid inside of this protein coat. Doesn't have anything else. Sometimes viruses will also bring with it enzymes. So for example, if we're talking about an RNA virus, we don't have or prokaryotic cells don't have any enzymes with the ability to take RNA and convert it into DNA, because that goes against the central dogma of biology. So viruses that contain RNA need to incorporate in its capsid, along with the nucleic acid, a special enzyme called reverse transcriptase. And that enzyme can take the RNA and put it back into DNA. But this is kind of showing you what it looks like. Okay, so they have a huge genetic diversity. So like I already mentioned, viruses can either contain DNA or RNA and never both. But in addition to having two forms of nucleic acid, they can also either have DNA or RNA either as a single-stranded form or a double-stranded form, or they can even be linear or circular or even segmented. So a good example is the flu. So influenza A virus actually has 8 separate RNA segments, but something like poliovirus is really just a single linear strand of RNA. So there's a huge amount of diversity that we'll talk about later on in this chapter. RNA viruses, though, in particular, these can mutate fairly quickly because their RNA-dependent RNA polymerase lack the proofreading ability. So RNA viruses typically undergo a lot of errors during replication, and this is why these types of viruses, like influenza and coronaviruses, often produce new variants. So we saw that during COVID-19. DNA viruses are a little bit more genetically stable. So this is one reason why some viruses, like pox viruses, have historically been more controllable through vaccination. Okay, so one of the most interesting things about virology is really host specificity. So most viruses can only infect a particular cell type within particular host species. So the specificity is determined by what we consider a lock and key interaction between the viral surface proteins, so those on the capsid or the envelope, and the specific receptors that are actually on the host cell membrane. So for example, human immunodeficiency virus, or HIV, it specifically targets the CD4 cells and CD4 T cells in humans, where something like West Nile virus can affect many different animal species, including birds, us, so humans, and even horses. So viruses are going to be classified as specific when they only infect one type of host cell. So HIV infecting human immune cells would be an example of a specific. While if they're considered or classified as a generalist, they would be able to infect a range of host cells. So like the West Nile virus that I just talked about. Okay, so viruses infect all kinds of organisms, but their distribution is a little bit uneven when we're talking about infection. So bacteriophages, again, these are viruses that infect bacteria. These are really, really, really abundant, and they actually outnumber the number of bacteria that we have themselves. So phages play really big roles when we're talking about regulating microbial populations. They're even involved in cycling different nutrient sources, and they are thought to influence microbial evolution. Plant viruses, they're a little bit, they're really common, but they're less well understood than bacterial or animal viruses themselves. So plant viruses are usually transmitted by an insect vector, such as an aphid, a leafhopper, or even a psyllid, something like that. Fungal viruses are really unique because they lack the extracellular stage that I was talking about before. They remain within the fungal cells themselves and then are passed to daughter cells during division and through cell fusion. So this is just showing you that sizes of virions kind of vary. So they range anywhere from about 20 nanometers to over 400 nanometers. And we actually have some giant viruses that are called Mimi viruses that you can actually see with a light microscope itself. But most of them are too small to be seen with light microscopes, and it would require an electron microscope in order to see them. Okay, so the capsid again is just a protein shell that is really just enclosing the viral genome, and it's just composed of subunits, protein subunits that are referred to as capsomeres. And these capsomeres can be made of a single type of protein or multiple types of proteins. And again, it has a couple of different functions. One, the capsid can act to help protect the viral genome, and the other function is really to help with attachment to host cells. Capsid structures are usually highly symmetric, which allows the virus to kind of build these stable particles without having to use a lot of genetic information, because their genome, although it's extremely diverse in what kind of genome it could have, it doesn't contain a lot of genes. So again, because it's going to use the host cell machinery in order to get what it needs. So there are three common types of capsid symmetry. So We could have something that's helical in shape, like tobacco mosaic virus. This was the first one that was actually discovered. We can also have icosahedral. So that was the one that I showed you on the previous image, with the capsid and then just the nucleic acid inside. Or they can even take on really complex kind of capsid structures. So viruses can also be broadly categorized by their shape. So they could be helical, they could be icosahedral or polyhedral, or again, they could be complex. So helical viruses down here, this is the example I just told you. It's kind of, one example is tobacco mosaic virus, or TMV. It was the first one that was discovered, it was actually discovered in a plant. And these have rod or filament-like structures, and they're formed by capsomeres, or those proteins kind of spiraling around the nucleic acid. Then we can have some polyhedral viruses like adenovirus, and these are kind of shaped like icosahedrons. There's one that even, well, this one in particular to me looks like the ball at the Epcot. You know what I'm talking about? Okay. Anyways, if we have this polyhedral kind of shape, it really is structurally stable. So this is the most structurally stable shape. Then we have our complex viruses, and a good example would be a bacteriophage. In this case, this is bacteriophage T4, the most well-studied phage that, in research. And they have unique structures. So they're also going to have things like a head, they have tails, and then they have these little fibers too that really help allow for precise host cell recognition and help it inject, kind of like a needle, its genome into the host cell. So its shape is also going to kind of influence how the virus attaches and how it enters cells, and how it is recognized by immune system, and also how it's going to be assembled inside of the host. And this is just showing you the complex shape at bacteriophage T4, and this is really a classic example. Okay, so many animal viruses, they're going to have the additional lipid envelope that we talked about initially. And again, this envelope is derived from the host cell's membrane during viral budding. So this envelope is not just an envelope. It does have things on its external surface. So it has viral glycoproteins. These are often referred to as spikes. So they're really essential for host cell recognition and its attachment. And the spikes are really specific, and they're going to determine which cells the virus can actually infect. And the envelope, again, is composed of a phospholipid bilayer and proteins. Where did these come from? Some are from the host, but some of the proteins are also encoded by the virus itself. So envelope viruses are usually more fragile in the environment than naked viruses, so those without an envelope, because the lipid membranes can be disrupted pretty easily by things like heat or drying or some sort of detergent to help break it up. So the envelope gives them an easier advantage in evading the immune system, but it's not good for survival outside of the host itself. Naked viruses, or those without the envelope, are generally more stable, but it's easier for the immune system to kind of detect them. So this is just showing you kind of what makes viruses, or what's unique about viruses relative to all the cells that we've kind of talked about before, and why they're considered a